So we were discussing reproduction bacteria, or like this was the non-sexual or not true sexual reproduction, conjunction, transformation, transduction. Conjunction was done, and even this topic is also called as mutation and genetic recombination. Well, I will just continue with the second step, which is transformation. Okay, let me write just first, otherwise this literally and second step. The second step named as transformation. I also have picture which I will show you later on. First you need to understand this topic. So transformation, simply a bacterium, like here you will see the bacterium or DNA would be taken, and this DNA would be taken from where? From our environment. Even the DNA could be shaded or like covered by other bacteria as well. So this means we are going to do this all experiment in laboratory. So the DNA which we are going to take will be introduced, of course, if you're doing in laboratory, so this will be introduced by scientists, and you will see that this then what you will observe that scientists will tell you a few things, how does the DNA looks like, or these, like if the DNA is in form of circular DNA, like if that structure is circular DNA, how is this the like not donor, of course that was in conjunction, but like this the cell. So you will see if the DNA is in circular form, so this would be like DNA in circular form, that's called as plasmid. Okay, that will call as plasmid, and this can be even copied. This means they can copy themselves, and they, when it will be copied, so they can receive, they get an ability to receive other cells and pass to the other descendants, which means like they can easily, this is plasmid if they are, that's in circular, if they're circular DNA, so that's called plasmid, and that plasmid can copy themselves and that can transfer from one cell to another. This was simply about transformation. We don't have much more information in our book. Well, this was the basic information. Now let's see in this picture. It's like how is that all. So this is the picture. First thing, I cannot write, sorry for that, but you'll see that this is circular DNA. When plasmid, a little structure will go inside, so like uptake of DNA competent bacteria, so it's like a little bacteria have been taken. Then what you will see, that this part will get attached with the genetic material DNA, but in later on in second part you can see a round structure have been formed, and that round structure is nothing more but it's like plasmid. So DNA may integrate into genome or exist as a plasmid. So both condition can happen, like the DNA can be integrated in the first picture you can see, or they can also form plasmid. So this is about like the transformation. I'm sure it's clear to you. This is not very detailed topic in our book, whereas now we are going to continue with the third step. This third step is transduction. So here what will happen, this simply a process where genetic material will be transferred. Genetic material is transferred. When genetic material is transferred, like this will be transferred from where to where, like they will be transferred from phage of two bacteria, between two bacteria. That simply means transduction. For sure you can get this very easily in the picture, so I'm sharing it there. So this is the picture. You can see the simply picture where genome would be entered first. Secondly, you will see that here DNA, like you can see in this pink pink is structure called viral DNA, and blue is bacterial DNA. In first picture, you can see DNA is going to insert viral DNA. Like this genome viral DNA would be inserted. In the second picture, you can see they both get separated. In third picture, you can see 4 other genome are found, and even few are going to exit or few are coming. This type of things happen, and they both have, like these all will have both material viral DNA and bacterial DNA. In fourth picture, just the same. This is like two processes which have been shown. In fourth picture, you can see the again going to insert and inserting both, like now they're inserting both of these structure, bacterial DNA and viral DNA, into one single picture. And the last, you will see that a few more a little wider structure being made around like a structure. This is showing that the genome, that viral DNA have been inserted. So I'm sure it's clear to you, and I'm just giving overview of this construction as well. This was simply a picture where chromosomal DNA was forming plasmid. It was transferring through pilus, which is the also called as cytoplasmic bridge. So these all process happen, and in the end, you will see two different cells of the new donor would be formed, and old donor would be also present. So here our this topic ends. Thank you for watching. Bye bye.